Photosynthesis is the process that plants use to create simple sugars. This equation right here shows what photosynthesis does. 6 carbon dioxide molecules and 6 water molecules, plus some light, will give you a simple sugar and 6 molecules of oxygen gas as a byproduct. In the where photosynthesis happens, chlorophyll A, chlorophyll B, and carotenoids actually collect the energy. These are pigments, and they accept light energy, and their electrons excite, and then the electrons are taken down the electron transport chains. So chlorophyll A is the most used one, and that takes in red and blue light, and it reflects green light, which is why leaves are green. Chlorophyll B takes blue and yellow light in, and it reflects blue-green light. That's also why leaves are green, because chlorophyll B is the second most used pigment. And carotenoids take in all of the energy that chlorophyll A and chlorophyll B would be harmed by, so it takes in green and blue light, and it reflects yellow and red light, which is why in the fall time, leaves become yellow and red, because the carotenoids reflect them. So, I drew a picture of a leaf. I'm the best drawer I know. I'm better than Mr. Jennings. I'm better than everyone. But this shows what a leaf is, and what's inside of an actual leaf. So over here is the waxy cuticle. It's on both sides, and it's secreted by the epidermis. But what the waxy cuticle does is it holds the stomata, and the stomata are what allows gases to actually enter the leaf. The epidermis are the guard cells of the leaf, so they protect it from bacteria and all sorts of other organisms that would normally harm the leaf. These cells right here on either side are the mesophyll cells, and that's where photosynthesis actually takes place. And this vein right here is where all the nutrients and water will come into the cell, or into the leaf, so the cells can use it. And all this space in between these mesophyll cells are where carbon dioxide and oxygen are stored for the leaves, or for the mesophyll cells to use, where they'll exit through the stomata, or enter through the stomata. Okay. So chromatography, for that part of the lab, the paper is cellulose. So as the solvent reaches the mixture of the spinach pigments that we took out, the nonpolar will go up more, while polar binds to the cellulose more. And molecular mass and solubility also affect what goes up the paper more. RF, the RF value, which is what we found, known as the retention value, was determined for each pigment. What this will show is it'll show which pigment went up the furthest, because it's the distance of the pigment traveled divided by the distance that the solvent traveled. For chlorophyll A, this is 0.04. For chlorophyll B, this was 0.027. For xanthophyll, this was 0.067. And for the carotenoids, it was 0.933, which shows that in order of furthest to it didn't move at all is the carotenoids, the xanthophyll, the chlorophyll A, and the chlorophyll B. In the next part of the lab, the spectrophotometer was used to measure the transmittance of light as a percent. DPIP accepts the electrons that will be given off by photosynthesis, and that's what spectrophotometer is going to be measuring. We had to find this to figure out which solution would be performing photosynthesis the fastest. The light reactions is the first step of photosynthesis. This occurs in the thylakoid membrane. Sunlight is taken into the pigments in the leaf, which their electrons get excited, and they're taken down the electron transport chain. The photosystem 2 makes ATP and supplies electrons to photosystem 1. Photosystem 1 makes NADPH. Water in this is split to make oxygen and H+ atoms, and H+ goes through ATP synthase to create ATP. And oxygen combines with another oxygen to make a gas and exits through the stomata of the leaf. In the Calvin cycle, which is the second part of photosynthesis and the last part of photosynthesis, it occurs in the stroma, which is outside of the thylakoid, which is where the thylakoid is in. RUBP accepts carbon dioxide, and Rubisco does this. It adds RUBP and carbon dioxide together. The next part of the Calvin cycle is where G3P, 6 of them, are created, and 1 is taken out of the cycle. And then the final part of it is where RUBP is regenerated from the 5 remaining G3P molecules. So in order, this is carbon fixation, which is where carbon dioxide is added to RUBP. reduction, where G3P is created and then 1 is taken out of the cycle, and regeneration, where RUBP is then remade from the 5 G3P molecules.